following what the presentation or the work that we're doing, we would ask that you uh, type in your questions on the chat box. And this way we will be able to see them and we will try to get to them at the end of this session and answer those questions to the best of our ability. Of course, a big thank you needs to go to all of the farmer cooperators that are willing and have worked with us in this past year in allowing us to come to their fields and work on their farms in putting these applications of the soil biology products, allowing us to come in and follow up doing testing and inspections, and also where we have a treated area and we have an untreated area that we can study and look at any differences that we can start establishing. We feel this is one of the largest on-farm soil health studies that we are aware of. We're covering an area of 4 different counties in Northwest Ohio, 29 different plots. So we have over 500 acres that is on the treated sections, and then roughly 500 acres on the control that we are monitoring throughout the course of the season. So we thank you for allowing us to do this. Without your support, we wouldn't be able to do these types of trials. We also need to thank our company supporters, the Douglas Plant Health people, Agri-Energy Solutions, and CanGrow Solutions for their support in helping us provide some of the financial efforts on the products and also helping us on some of the testing fees that are incurring. Without their help, this would not be possible for us to do this on this kind of a scale that we're doing it. So the purpose and objectives behind this project: soil health and plant health. This is something that in the past 3 to 4 years, we are just hearing a lot of new effort, new financial investments by companies and by a lot of people in approaching this thing of what is soil health and what do we need to do to improve our soil health. There isn't an agricultural company hardly anymore that has not come up with their own products, their own solutions. And there are many, many different efforts out here of approaching this thing of soil health. When we started this about 7 years ago, how we can approach the improving the efficiency of the products that you are a farmer applying to your fields. This was our initial effort. How can we make nitrogen or phosphorus more efficient and get better performance from it for the money that's being spent? And how can we use soil health to improve our crop yield? Another whole element of this has been added recently in looking at soil health and how can it be used to improve our water quality, and also how can it be effective in improving runoff of nutrients from our farms and fields, especially things like phosphorus for our Lake Erie watershed. So soil health has also now become involved in looking at, can it be one of the solutions in improving our whole water quality issue? We're now seeing that the government's programs are getting involved through things like NRCS, H2 Ohio, to follow and encourage using very specific treatments of application of product, certain cultural practices as a way to improve water quality, and soil health is one of their areas that they're working on. Most of the work that NRCS, H2 Ohio, as well as the universities approach to improving soil health is the use of cover crops. There's been a lot of work or emphasis being put on the need to utilize cover crops as a way to improve our whole cropping and water quality issues that we uh, are dealing with in this area. Uh, to get consistent results in managing cover crops, there's been a lot of work being done. Farmers have done a lot of work. There's been some progress made, but the issue of getting good stands of cover crops in our Northwest Ohio region, in our corn and soybean rotations, is requiring quite a bit of additional management, and it's also requiring sometimes some significant new investments. So is this the whole answer? And this is the question that we have approached in these trials. Is using cover crops our whole solution, or is it the only solution that we have available? Getting good consistent stands and managing cover crops while at the same time we're trying to grow our cash crops of corn, beans, even wheat has become the question that has become the challenge, I guess you'd say, that we have out here. The objective of our field trials is what we're trying to do is see if we can take the crops that we are already growing, mainly the corn and soybeans, and can we utilize those crops to try to improve our whole soil microbiome biology, the whole soil life. You want to take the next slide? We know that, there's certain elements that all are required. We do have to manage our physical condition of our soils through drainage, how we manage working our soils, uh, the nutritional side. We've spent a lot of years working with what is the right amount of fertilizers do we need, what is the nutritional needs of our crops. And we now are just really starting to really delve into, and a lot of different people are, what is the biological element of our cells that we need to address to make all of these things come together. You take the next picture. One of the things that we are approaching and what we're trying to emphasize in how can we use the existing crops we're growing. We know that in a plant that it requires basic things like water, it requires sunlight, and it requires air, oxygen, and it also requires a certain mix of the right kind of minerals and nutrients. But all of our energy that we really use in our food is coming from plants, and it comes from this whole process of sunlight producing sugars and energy, amino acids in our plant leaves. And the way a plant works that we are growing is that roughly 50% of that energy that we are getting from sunlight and the photosynthesis is used to grow our plant, to grow the more leaves, to grow the structure, to be able to store enough energy in the plant to then reproduce and grow the grain and fruit, whatever that plant produces. But 50% of this total energy that we are manufacturing in these plants is moving down to the root system. And here is what makes our roots grow. And we do have to have enough energy to expand our roots, to be able to continue to be able to get enough water, other nutrients, and so forth to uptake to feed the growth of this plant. Also, of that 50% that goes to the roots, roughly 25% of that is released out of the root system. And this is the area that we are addressing because that energy, that sugars that is released out of the plant roots is what feeds the microbes in our soil. And that's the food source because these microbes, bacteria, fungi, algae, protozoa, these are all living organisms and they need food. And the food that they get is from what the plant is producing through photosynthesis. What we are asking and trying to do is apply a whole additional amount of a broad spectrum of different microbes while this plant's growing, and allowing the plant, whether it be the corn plant, the bean plant, wheat, to feed those microbes and increase the amount of biological life that we have in the soil. So this is the approach that we're looking at, that we're testing for, and to see if this is also another tool that is available for us to enhance and build up our soil life to make our soils more efficient. Going to take the next slide. We know that our soil is a living organism. It's not a piece of dirt, of clay or sand. It's made up of billions of different types of living organisms. And the broader spectrum that we can make of this, of the different kinds of bacteria or the different kinds of algae, the healthier this soil will be, the more it can resist stress and diseases and so forth for growing our plants. Next slide. So soil is more than dirt. It's a microbial activity that we are trying to enhance around the plant roots, which is called the rhizosphere, where these microbes live, and they break down a lot of the nutrients that we need. They convert the phosphates, the nitrogens into usable form that the root can take up and get it up into the plant for use with photosynthesis. Next slide. So soil and the benefit of improving its whole structure, the ability to make the soil more granular, the ability to have a better way that the roots can grow and live healthy in the soil is a big part of what soil health means in what we're trying to accomplish. Next slide. Water penetration and water use is one of the big factors that we are testing for. Can we improve the ability of our soils to absorb water where it doesn't run off as quickly, and also hold water so that it doesn't go down through the soil into our tile lines, and it has more ability to act as a sponge, if you will, in holding water along with the amount of oxygen and other things that soil needs. So this is what we're testing for. And so what we'll do is turn this over to Marty Huffbauer, who's going to start providing some of the information that we've gathered here in the first year. And we'll be going through the tests and everything that we've run and a little bit of the results. So thank you. And Marty, it's yours.
Marty, you're still muted.
All right, thanks. All right, thanks, Les. Good afternoon, everybody. Just an update. In total, we had 29 total plots this year, 10 of them being soybean, 17 being corn, and 2 of these plots being wheat. Obviously, we were scattered over a wide range of areas, and I think 4 different counties we were in total. Can we go to the next slide? So a quick refresher on what our rates were, and just so everybody knows what the cost of these products are. And going forward, for the broadcast applications, the rate was 2 gallons of the SP1 and a quart of BioLine, and that comes to about $18.10 an acre. For these 2x2 and in-furrow applications, we did 1.5 gallons of SP1 and a pint of BioLine, and that comes up to about $11.95 or $12 an acre. If you go to the next slide. So for our soybean plots, we had 6 minimum till or no till plots. 4 of these soybean plots were with cover crops. So that gave us some more data points to kind of evaluate if we're getting cover crop like benefits here. 4 of these were conventional tillage, and all of our soybean plots were broadcast. For corn plots, we had 12 strip tilled, minimum tilled plots, 4 in furrow, 5 conventional tillage. And for application, 11 of these were broadcast and 2 of them were applied in 2x2. So all this kind of leads to the next question would be, what did we do to, measure the difference of the product that we were using? We had a nice variety of different tests, and complexity is a little different of all of them, but Keely will elaborate a little bit more later on the Ward Laboratories Haney test and Biomakers. They kind of take the DNA of the soil, and like I said, we'll get into that a little more detail later into the presentation. And we had some fertility tests as well as soil respiration. You go to the next slide. We also took visual observations mid-season and late season. Root digs, took photos, observations of the ear size and pods late season. Another measurement we took was a simple penetrometer reading to show us where the compaction layer is and if we're having any effects on those compaction layers that may be out in these fields. Next slide. So you can see some of the observations we took with the root digs and ear checks, things like that, yield checks. And that was one of the measurements we took for the next slide. So one of the tests we did across all the farms was USDA soil health card. This one is a simple visual observations. You can see here there's score 1 to 3, 1 being the lowest, 3 being the highest, and each of these categories we score each of them, and at the end we'll have a total score. Soil structure and tilth was one of the categories. Another category we took would be, you can go to the next slide, soil life, things like earthworms, the earthy smell you look for in a healthy soil, how well is residue being decomposed out in the field, things like that were all scored in each farm. There's also tests on soil and water, things like drainage, water holding capacity, water movement, things like is it running off, is it soaking in, things like that nature. Next slide, please. Another observation that was on this card is overall plant vigor, plant health. You can see uniformity and growth, seedling emergence, root systems, all of those again were scored 1 to 3. And then finally, fertility. So yeah, fertility management. So the soil test levels we did have, we scored those there on nutrient levels, soil pH, and organic matter. And you can see there at the bottom left there, you get a total score, and that'll give us a range out of 45 there. So next slide. And then at the end of the season, we did yield checks. We had a good bit of growers that had yield monitors. So obviously we pulled that in and analyzed it through our software here. And then the remaining plots, we did weigh wagon tests. So we obviously went out in the field and took our own observations and calculated what the yield was based off of that. So with that, I'm going to pass it back or over to Keely, and she's going to explain the Haney test a little better and some of the other tests we took. Thank you.
So like Marty mentioned, we took a variety of different soil health tests this summer. This first one that I'm going to go through is called the Haney test, which is administered by the Ward Lab. You can see that there is a lot of numbers on this test, and some of them might be familiar, like soil pH or organic matter. But there's a couple of numbers on this test that I like to pick out and focus on. The first one is this soil respiration number. So this is actually a measure of how much CO2 is being released by these microbes. So just like when we're active and exercising, we're always actively releasing CO2. And the same is for the soil microbes. As they're moving throughout the soil and doing their various functions, they're releasing CO2. So this is a measure of how active those soil microbes are. The other calculation I like to take a look at is this nitrogen release and nitrogen reserve, and there's also values for phosphorus as well. But this nitrogen release number is the amount of nitrogen that those microbes are actually working to release. So after they move through the soil and mobilize those nutrients, this is the amount of nitrogen that they release. Whereas the nitrogen reserve is how much is left in that pool after the microbes have done their job. And then the last thing that I like to pay attention to is a soil health calculation. So this test takes into account all of the numbers that are on this sheet and gives you a soil health calculation. This is on a scale from 0 to 30, and according to the Ward Lab, a 7 would indicate a good soil. So in this case, we have a 9.43, which would be better than a good soil. So now moving into another test that we took this summer, this is a biomarkers test. So this is the summary page that you first see. On this page, it actually gives you how many different microbial species are in the soil on this particular farm. So here we have 671 different species. And then it further breaks it down into whether those microbes are fungi or bacteria. And then it breaks it down even further into more specifically what those microbes are. So one of the first calculations we have is the biodiversity reading. So this refers to how many different microbes are throughout the soil. So in this case, there's a high diversity, meaning there's a lot of different species. And then there's also a functionality score. This refers to how well these soil microbes are able to do their job. And like we mentioned before, one of the jobs that these microbes can do is actually mobilize nutrients. So this functionality score is important to pay attention to in that aspect. And then the final score on this page is a resistance reading. So this refers to how well these microbes are able to withstand stress. And this was an important reading, like for this summer when it was really hot and dry, were those microbes able to withstand those harsh weather conditions? So not only does this test detect beneficial bacteria and fungi, but it also detects harmful pathogens as well, and kind of gives you a watch out list of what was detected in the soil and what you should probably be scouting for or keeping your eyes on. So a couple things on this list that are probably familiar: anthracnose, fusarium, even some pythium. And then it also gives you the risk level as well. And it also lists all of the pathogens that were not detected in the soil. So this biocontrol reading actually groups the microbial species based on if they were to encounter either a disease or an insect or even a nematode, how well would those microbial species be able to assist in fighting off those diseases, insects, or even nematodes. And then this hormone production reading, it groups the microbial species based on what hormone they're capable of producing. So in this case, this farm had a high auxin production reading, and it even lists what auxin is important and what job does it do. And it's important in cell division and stem elongation. So now the stress adaptation, they group the microbial species based on what metabolites they produce to withstand stress conditions. So just a quick reading, this one was high in abscisic acid, ABA, which is important in growth regulation, plant resistance, and increasing yields. This is one that I think we found particularly interesting. So like we mentioned earlier, one of the jobs of microbes is to mobilize nutrients that are in the soil. And this actually gives us a reading on how well those microbes are mobilizing different nutrients. So the microbes in this soil are doing a really good job of mobilizing carbon, not so much on nitrogen, but they're doing an okay job on mobilizing the phosphorus and potassium that's there. So I thought that this was also a pretty good indicator of how active the microbes are in the soil based on how much nutrients they're mobilizing. And then this finally just gives us a reading on where we're at with our minor compounds. So now we'll move into some results to analyze these results. And today I'll be focusing mostly on stand counts and yield data. We broke these up based on kind of what agronomic system they fell into. So we compared things such as conventional tillage soybean compared to no-till soybean, fields with cover crops, fields without cover crops. So they're broken down to be a little bit more similar. And for all of these graphs, on the x-axis, we'll have our treatment. So we'll be comparing the control area to the treated area that had SP1. And then on the y-axis, we'll have what we are measuring. So in this case, plant population, or on this graph, it'll have average yield. So we'll start with the average stand count, and this is for a conventional tillage soybean system. As you can see, the stand count was pretty similar. And for all of this data, we only ended up with one system that has significantly different stand count. So we'll go over that in a couple of slides. But as you can see, pretty similar as far as the stand count goes. But once we get over to yield, we saw a 5.9 bushel increase in the treated area compared to the control. So now moving into minimum tillage soybean. This is where we saw the significant stand difference. So as you can see, the control had about 90,000 plants, whereas the SP1 treated area resulted in about 108,000 plants. So that would be a significant plant population difference. However, this didn't exactly result in a yield difference because we saw about a 1 bushel loss in the treated area compared to the control. So that 1 bushel loss, you're kind of splitting hairs, but that was the difference we saw. So now we'll look at soybean with cover crops. Again, you can see that the stand count is pretty similar. It wasn't statistically significant. And we also saw that about 1 bushel yield loss here as well in the treated area compared to the control. Now for soybean without cover crops, similar story. We're still not seeing a statistically significant stand count, but we're seeing a 5.9 bushel increase in our SP1 treated area compared to the control for soybean without cover crops. And now to get a reading for all of our soybean data, we pulled it in regardless of the agronomic practices and just looked at it on a per crop basis. Again, very similar stand count, but we did see overall a 2.5 bushel increase in our SP1 treated area compared to the control. And again, this is for all of the soybean fields that we had. So now we'll move into some results for corn. So since we had several different application methods for corn, we broke it down based on application method as well as tillage. So for this first one, this is for broadcast applications on corn. We're seeing, again, a very similar stand count. I'm sure you guys are gonna get sick of me saying that. But we did see a 4.7 bushel yield advantage in SP1 treated area compared to the control. So now for the in-furrow application on corn, again, a stand count that is not statistically significant, very similar, but we are seeing a 2.2 bushel yield advantage in our plots where the SP1 was applied in furrow compared to where the control was with no SP1 in furrow. So now for SP1 that was applied in the 2x2 on corn, again, a very similar stand count, but it did result in a 4.9 bushel increase in the SP1 area, the treated area, compared to the control when SP1 was put in the 2x2. So now for conventional tillage corn, again, a very similar stand count, but we did see a 2.9 bushel increase in the SP1 treated area compared to the control. Okay, so now for strip-tilled corn. One thing that I found pretty interesting, out of 17 corn plots, 12 of them were strip-tilled. So that was pretty cool to see that that practice is so popular. A very similar stand count on those strip-tilled cornfields, but we did see a 4.6 bushel yield advantage in the SP1 treated area compared to the control. And then finally pulling all these cornfields together to get an overall picture. Very similar stand count, but overall in our cornfields, we're seeing a 4.1 bushel yield advantage in the SP1 area compared to the control. So one of the things that we didn't have the opportunity to look at this summer because it was so hot and dry was disease. So now Doug is going to discuss a little bit about some past research that's been done as far as SP1's effect on disease.
Yes, good afternoon. I wanted to briefly touch on some other observations that we've made in the last few years since we started testing SP1, mainly in the area of disease control. In vegetable crops, we've seen a reduction of soil-borne diseases like Phytophthora, Rhizoctonia, Pythium, Verticillium, Fusarium, and Sclerotinia. It is known that many of the organisms in SP1 cause a increased systemic acquired resistance response in plants. It seems that when the concentration of the good microbes outnumber the bad ones, the plants are able to fight off these diseases. In corn trials, there was a noted reduction in ear mold. This first slide shows observations made by Dr. Ron Henniger at North Carolina State University, and showing a difference in infection in the grain filling period. There was several different years when evaluating the corn plots during grain filling and finishing stages, we've seen a marked reduction in Gibberella ear rot. Next slide. This is also some observations made by Mike Netz locally in trial plots that he put out, also showing a marked reduction in ear mold. Of course, Gibberella ear rot is a major concern because the fungus produces the mycotoxins, including vomitoxin, that are harmful to livestock. I believe that's all I've got. I'll turn it back to Keely.
Okay. Maybe I'll get this right. All right. So to wrap up our results section, we did take penetrometer readings this summer, like Marty mentioned, that measure compaction, but they were pretty inconclusive because a lot of times when we went out to the fields, it was so dry that it was really hard to actually even get the penetrometer in the ground. So that was pretty inconclusive this year. But 76% of our plots that we had out had a positive yield impact in the treated SP1 area. And that ranged anywhere from a 1.3% bushel increase to a 25% bushel increase. When we did our root digs and visual observations, we saw that there are much larger and more extensive root systems in the treated area in our in-furrow and 2x2 plots. And you can see that over here to the right. This is a photo from one of our root digs done in a 2x2 plot. On the left, you can see that the treated plant has a much more extensive and bigger root mass compared to the untreated plant. We do think that the hot and dry weather played a role in affecting our broadcast applied plots. As soon as we got those plots applied, which was right before planting or soon thereafter, the rain shut off, it got really dry. So we're not sure if that product ever really got worked into the ground the way that it could have. So in the future, we're going to begin year 2 of 3 of this SP1 study this spring. We're planning on implementing new observations and tools along with repeating what we've already done. We're planning on using a soil moisture reader, which is one of our tools we didn't get to use last year. And then we're also going to be implementing a water infiltration test as well. One of the unique things with this SP1 study is we asked all the growers to just keep doing what they're doing. Don't change any of your agronomic practices for the trial. We just want to introduce SP1 to your farm and see what it could do. So that gives us the opportunity to analyze more diverse agronomic practices. And then Les has an update on SP1 now being MVP.
Okay, Keely. Just want to mention that the product we're going to be putting on this coming year using is going to have a different name. And we just want to assure everybody that it still is SP1. What's happened is that we have a change of ownership. The company that we're working with was purchased, and it's still manufacturing the same products, but the new name is going to be called MVP. So when you see that, when we talk about putting on MVP, it's going to be the same biological makeup as what the SP1 was. A couple other things that I will just wrap up here and mention. A lot of the tests, like what Keely mentioned and so forth, this coming year, we are going to be doing and showing a lot more where we're running those tests on the treated area and then comparing those to the control. So that we will now start looking at what the difference of maybe a Haney test would be, or a test of the biomarker, where we will start comparing the treated zones compared to the untreated zones. So not only yields, but some of the tests that we're going to run, we will start showing, is there a difference showing up? Is there something that we're being able to quantify that we are changing in the soil makeup itself? Another area is we are continuing to look at ways we can enhance this whole biological activity. And when we're putting on these organisms, as we mentioned, they need a food source. We're taking the plant that we're growing and using it to feed those microbes that we're applying along with what's already in the soil. And we are looking at several different products that maybe would add some food sources or start getting those activities of organisms going quicker. And one of them is a product called Synergy. And what Synergy is a food waste product. We have tremendous amount of food in this country that is being thrown away from our grocery stores, from our restaurants, where they're taking, companies taking these food items that either are spoiled or have run out of their due dates to be able to be sold. And they're taking this food and putting it into a mix and standardizing the ingredients. And it's a very high form of amino acids, sugars, of course, the nutrients that we have on the food that we eat. And we're utilizing that to see if we can add the activity that we're looking for of these microbes that we're applying, plus what's already in the soil. There's been some pretty good work already showing that there is a pretty good activity benefit. So that's just one of the items that we are continuing to look at in this whole effort to try to enhance our whole soil life, to improve nutrient efficiency, to try to get a better return of investment to you, the farmer, on the crops that you're growing. So with that, we will wrap it up. If you do have any questions, we will look to see if there's any questions been asked, and we will try to answer them. Did we notice any questions, Keely?
Yeah, if you have any questions, you can go ahead and type them into the chat box and we'll read them and answer them. We'll go ahead and give you guys a couple of minutes to think up some questions and put them in the chat box. In the meantime, Doug has some comments that he wants to make before we close.
I did want to emphasize that we as a company are going to continue to explore ways to offer services and tools that have practical applications for growers on their own operations. We can point to this project we're in the middle of as an example by trying to establish some benchmark indicators of soil health and then seeing if we can influence those indicators by increasing the populations of good soil organisms. We hope we can advise on some systems that have a positive impact on your operation without being disruptive. A new system or a product that gives a big response but is impractical or cost prohibitive is of little value to us. So despite what appear to be yield responses, even in this first year, we feel that if we can affect measurable changes in the desirable soil properties over time, that a direct and consistent yield response will result. Thank you.
Okay, so it looks like we do have a question. The question is, how many of these plots had 5% organic matter? The slide for healthy soil said it should have 5%. Is that normal for this area?
I'll take that one, Keely. We have very few of our soils overall that are 5% or more of organic matter. We do have a lot of 3, 4% sometimes. We have some in the lighter soils that run down to 2.5, but we don't find, there is a few pockets, but we don't find very many that are up in that 5% or more range of organic matter in our lake bed soils.
Okay, we don't have any more questions right now, but I will keep the video and the chat open for a couple more minutes. Oh, here's another one. It says yield increases were not fabulous. Additional root growth is definite. What else can we do to get the tops of the plants to match the improved roots? Were tissue tests taken of the ear leaves during grain fill?
You want to take that, Keely?
Yep, we did not take any tissue tests this year. That wasn't part of our testing.
And the other thing we want to mention is we're not expecting, or were we promoting that we're going to see this big yield increase the first year. This is a progressive trial. We're not going to build up this biological activity in our soils with one application in 1 year. We didn't get to the diversity we now have in our soils overnight, and nor are we going to change our soils with just single applications. That's one of the reasons we're working on 3 years. We hope that over a 3-year period, we can start seeing some trends, and we can start seeing some things that become justified. But we're sort of actually pleased that we were able to show some yield differences, but we're not looking at that as being either our sole thing that we're trying to measure, or do we expect that to be something until we get further down the line with some of these trials. There's a whole series of water penetration, utilization of moisture, mitigation of stress. These are all things that we're going to be trying to measure. So hopefully that helps answer. Well, thank you, everyone. And certainly if there are questions, you can always contact us, any of us here at WNA. There is another question that came through.
Yep. It says, I think we saw a reading for ABA in one of the biological measurements. Isn't that normal to have a higher reading of ABA in hot, droughty conditions?
The answer to that is yes. That would be true.
Mhm.
Okay. Again, appreciate all the help from the farmer supporters and everyone. We will certainly be wanting to continue this effort for another year and appreciate all the help that we are receiving. So thank you.